Hello, everybody, and it's fun to be back at Pond Family Day. So before I start, show of hands. Who here is here because either they are a man with a neurodevelopmental condition or their son has a neurodevelopmental condition? Now, who's here either because they're a woman with a neurodevelopmental condition or their daughter has a neurodevelopmental condition? So that's about the ratio that you would expect. So for those of you who didn't raise your hand for the second question, welcome to the coolest club in the world. They get rid of slimy girls. For those who don't know Calvin and Hobbes, my apologies for the Calvin and Hobbes cartoon. But it's one of the things that as we're thinking about any of these conditions, it is remarkably striking what a role sex has to play in them. So that'll be the focus of my talk. It'll be a bit of the sex differences in neurodevelopmental disorders in neurodevelopment, and with quite a bit of a catch-up on the basic biology of sex differences in the brain and in brain and behavior. And then at the end, touching a bit on the impacts of that in research, and maybe with some allusions to clinical practice as well. But mostly it'll be on the basics of sex differences in the brain. But first to start. So if we look at sex differences across mental health, and this is not just pediatric, just about every single mental health disorder has a skewed sex ratio in one way or another. So if you look at depression, it's about 3 times as likely in girls or in women as it is in boys or in men. If you look at autism, it's about 4 to 1, and we'll get more into the details on those numbers later on. Attention deficit disorder, somewhere between 3 to 1 to 5 to 1. Multiple sclerosis, about 3 times as likely in women as in men. Alzheimer's, about twice as likely in women as in men. Obsessive compulsive disorders is one of the few that's actually at a 1-to-1 balance. But even there's an interesting difference in timings. That earlier onset OCD tends to be more in boys, and later onset OCD tends to be more in girls. Schizophrenia is about 1.5 times to 1, and you could go down these lists. And I'm not even mentioning some of the most widely skewed ones. If you look at eating disorders, they're somewhere around 12 to 13 times as likely in girls as in boys. So this is remarkably striking. And most of the time you don't hear sex discussed all that much when you're talking about these disorders. But effectively, one way to think about mental health and mental health disorders and conditions is that in order to understand them, you really have to understand these ratios. The risk of having autism, if you're a boy, is 4 times as much as if you're a girl. There's very few other things we can point to other than individual genetic mutations that are anywhere close to that kind of risk ratio. Now, in looking across some of these ratios here, there's a few commonalities that begin to pop up. If you're thinking in terms of psychiatric terms, the disorders that are more common in boys than in girls, or men than in women, tend to be externalizing disorders, so conduct disorders type disorders, whereas the ones that are more common in females tend to be internalizing disorders, such as depression is the greatest example. Also, if you look at the timing of it all, most of the disorders that are more common in boys than in girls tend to have their onset before puberty. So autism, ADHD, et cetera. These are all early childhood onset disorders. Most of the ones that are more common in girls than in boys or in women than in men tend to have post-pubertal onset. This is especially the case for all of the childhood mental health conditions. So if you look at depression, it's very rare to diagnose depression in a young child. It's much more common in adolescence. Eating disorders, much more common in adolescence. Autism, ADHD, much more common to have initial diagnosis before puberty. So before I get into the basic biology of this, just one little aside that this is in some ways a quite complicated topic, and I'm going to simplify it quite a bit. So go into some nice reductionist biology, and I'm really going to be talking about sex as biological sex. But of course, when we're thinking about it in a more clinical and more in human society, the spectrum between sex and gender is a wide and a complicated one, where very simply, the way that we tend to use the terms is sex purely corresponds to biological sex, and gender is the self-identification or sometimes the society's identification of gender in that case. And so sex tends to be considered fairly binary. I won't get into some of the rarer disorders where the biological sex can be difficult to differentiate, whereas gender tends to not be purely binary, where there's different types of gender identifications that aren't purely just male and female. And I'll be talking mostly about the physiology of sex differences, but of course, so much of what encompasses sex differences in society and in mental health has to do with the social construct of sex as well. I just wanted to throw it out there. I'm going to keep things fairly simple and talk about biological sex, but of course we have to acknowledge that this is a much more complicated topic than just the question of biological sex. But now into the basic background is, how does sex differentiation actually work? So, to begin with very simply, we all have 23, well, almost all of us have 23 pairs of chromosomes. And 22 of them are known as the autosomes, which are going to be the same in boys and in girls, and men and women. And then the 23rd is going to be either two X's or an X and a Y. So of course you get the X's from your mom. If you have two X's, you get one X from your mom, one from your dad. And if you have an X and a Y, the Y comes from your dad. So at the basics, in terms of genetics, the difference lies in whether you have two X's or an X and a Y. On the Y chromosome, and the Y chromosome, for those of you who want to study any genetics, is a runty little thing. It has many, many fewer genes than any other chromosome that we have. And there's interesting evolutionary arguments about selection for the Y chromosomes quite different than it is for selection of genes in any of the other, on the X chromosome or any of the autosomes. On the Y chromosome, there is a single gene called SRY, or the sex region on Y chromosome gene. And this one gene determines whether you will have biologically male or biologically female sexual organs. And so if you have the Y chromosome, you will have this gene expressed. The way it then proceeds from there is that early in development, and the timeline that you see here at the bottom is for rodents, but I'll get into the equivalent for humans in a second. But effectively, early in development, as the embryo develops, it has a so-called bipotential gonad. So the sex organs could become either male or female sex organs. And if you have the Y chromosome, it then begins to cause a series of different genes being expressed to then get testicular development at the end of that process. If you do not have the Y chromosome, so if you do not have the SRY gene, then you will go through the default sex development pathway, which is the development of ovaries rather than testes. Going a bit more across time, here you have a map of both the rodent up top in terms of timing and then the human at bottom. So again, you're born, conception at day 0, and then somewhere around the first trimester in humans, or about 10 days of gestation in rodents, this process of gonadal differentiation, so when you have the SRY gene, begins to occur. Then, in around the second or third trimester, or just a few days before birth in rodents, the gonads release testosterone, if you have male gonads. So that's what you see in the blue up here, is around birth, or in rodents, or in the second or third trimester in humans, there's a big surge of testosterone that goes through your system. And that surge is going to help determine the parts of your brain that are sexually dimorphic or that differ between males and females. They tend to be programmed early in this stage. I'll describe those a bit more in a second. Then if you have ovaries, a little bit later in that process, so just around birth, you have a surge of estrogen or estradiol that goes through the system. And then everything goes quiet. So the different hormones that are released by the ovaries or the testes, they go quiescent until you begin to hit puberty around age 10 to 13 or whatever it might be, where you then have another big surge of testosterone in boys, and then the cycling estradiol in women as they go through the periodic release of it. So again, there's really 3 key timing components to determining sex or biological sex in mammals. There's the initial development of differentiating the bipotential gonads into the testes or ovaries. That happens around the first trimester. Then there's the surge of testosterone that programs at least parts of the brain to be more male-specific or more female-specific. That happens in the second or third trimester. And again, it's always the default developmental pathway in the brain and in the body is the female pathway. It requires the presence of gonads and the release of testosterone to get the differentiation into male-specific organs, male-specific brain regions. Then everything goes quiet after birth. There isn't much hormonal release anymore. I mean, of course, it's not that there's nothing, it's just no big surges. And then again, around puberty, you have the next release of testosterone in boys, estradiol in girls, with the testosterone being constantly present in men and the cycling estradiol in women. So that's talking about sex organs. And then in around the 1950s or so, there was always, until then, there was the assumption that the brains between boys and girls, or between female rats and male rats, are approximately the same. And very coarsely, that's true. So one thing to keep in mind, and I'll reiterate that a few times, we're talking about a few fairly small areas of the brain that are very much specific to boys and girls, or to male rats and female rats. And most of it is going to be very much identical across sexes. But it was in the 1950s that they discovered that there's a few nuclei that are very much different between male and female rodents in this case. So one of them is the sexually dimorphic nucleus, and it's in the hypothalamus. You don't have to worry very much about what it does. But you can see it in these stains here, where all of the darker points are neurons. And you have one of the lateral ventricles, medial ventricle in the hypothalamus here. And then in males, so these are rats, you have this big area of lots and lots of neurons. And then if you look at female rats, you have this very small area that's corresponding to it. So it's about 5 times as many neurons in this area in the male rat as there are in the female rat. And remember how I was talking about this surge of testosterone that you have here. So in the rat, if you take this rat at birth, which is around when the surge begins to happen, and you give it a big injection of testosterone, so this is the experimenter doing it, you then get this part of the brain that looks exactly the way that it would in males. Even though the sex organs and all that are perfectly female, the brain begins to differentiate into what the male nuclei begin to look like if there's testosterone that's given to a female right around birth. So it's the sex organs differentiate, and then it's this extra release of the sex hormones around birth or second, third trimester in humans that begins to program the brain in male-specific ways in those few areas that are very sensitive to it. So the initial excitement about this was that it was all related to beginning to identify the parts of the brain that are responsible for sexual behaviors. But it became clear that that isn't just isolated to the classic sexual-related behaviors. And some of that work really came out of songbirds. I'm sure many of you who've been out in the woods and who've watched birds know that it's often the males only who do have complex songs, complex mating songs. And female birds in many species, not all, tend to have fairly quiescent singing or much simpler calls, simpler sounds. And they found one area of the brain, called RA, which is quite different depending on species and depending on the sex-different behavior in those species. So the zebra finch is one of the birds where the males have complex and fun to listen to songs, and the females have fairly simplistic songs. Whereas the European starling, both the males and the females have about equally complex songs. And if you look at this part of the brain, you see it here. In the male zebra finch, it's much, much larger than the equivalent in the female zebra finch. Whereas in the starlings, it's a little bit harder to discern, but the arrows point out the boundaries. It's approximately the same size. So again, some evidence across evolution that some of the sex-specific behaviors actually result in differential brain structures that seem to some extent, control these types of differences. So there's many of these nuclei throughout the brain. I'll go through some more maps in the area. And some of the interesting things about those parts of the brain that are sexually differentiated tends to be that it's not that there is no single structure that we know about in the brain that only exists in one sex. They always exist in both, but the amount of neurons or the size of them will differ by sex. And very often, the behavior associated with that nucleus will also differ. So for example, if you look at the medial amygdala, if you stimulate it in a male rat, you will get aggression towards other males. If you stimulate it in a female rat, you will not get aggression towards other female rats, but you will get maternal aggression towards pups, so only in a rat that has had offspring. So again, sexually differential behavior depending on environmental cues in that case as well. So that was all in rats. A bit of an overview of what it looks like in humans. So the first thing in humans that's quite striking to notice is that the male brain is about 10% bigger or so than the female brain. And that there's some differences in terms of development as to when peak maturation occurs. So if you look at that in the top plot here, so you have the females in red and the males in blue, the peak volume in total brain in females is around 11, 12 years of age, whereas in boys it's around 14, 15 years of age. And then you can begin to divide that up, looking at different parts of the brain, and of course this overall pattern of male brains are bigger than female brains holds. Now in some ways that is relatively uninteresting because that's a very simple, very straight relationship to overall size. And you see this across evolution as well. The bigger the mammal, the bigger the brain. So some of the blue whales have much, much bigger brains than we do. It's probably not worth arguing that the blue whale language or the blue whale tool use compares to what we can do as well. So there's no simple relationship between just bigger is better or bigger is smarter, but there's a direct relationship to overall size of the organism across evolution and to some extent within a single species as well. I'll go through it again a bit in mice, and the main thing I want to draw your attention to as I'm going through different slices of the mouse brain is less exactly where things are located. I don't expect you guys to be mouse neuroscientists by the time that this half-hour talk is done, but just to realize how much of the brain actually is sexually differentiated and how much of it is not. And again, it's fairly isolated. It's the parts of the brain that really seem to have sexual differentiation. So as I go through the brain, areas that will show up in red are bigger in females, areas that show up in blue, areas that show up in red are bigger in males, areas that show up in blue are bigger in females. And I can go through different slices, and there's clear isolated parts of the brain. So here you have the bed nucleus, clearly in one of the brain areas that's involved in social communication in males and in sexual interest. It's bigger in males, but most of the neocortex is either not different or slightly bigger in females. And the same kind of pattern holds as I go through the brain. There's isolated areas that are clearly differentiating male and female brains, but most of the brain is exactly the same between male and female mice or boys and girls. Now, of course, you can begin to play interesting tricks in mouse genetics to begin to understand where some of these sex differences are coming from. So until maybe the last 10, 15 years, the belief was that really everything that we know about sex differences is programmed by this early surge in testosterone. And so what matters is, do you have testes or do you have ovaries, and how does that impact brain development? But if you go back to the very beginning, there are differences that go beyond just the development of male or female sexual organs. Because of course, females have two X chromosomes, males have an X and a Y chromosome. And the next talk you'll hear from Michelle is on epigenetics, which is how you silence different genes as well. And so what happens in females is the one X chromosome is fully expressed, and the other one is mostly silenced, but there's somewhere around 5–10% of genes that are expressed twice in females. But of course in males you have an X and a Y chromosome, so there's differential gene expression, just because of the chromosomes that you have. And so what you can do in mice is begin to try to separate out what are the differences in brain development and or in behavior if you separate out this effect of having an X and a Y chromosome versus two X chromosomes and having male or female gonadal development. And so the way you can do this is you can take this one gene, this SRY gene, and you delete it from the Y chromosome, and you put it in again somewhere on an autosome. So what that means, you can have 4 core genotypes of mice. You can have mice that have two X chromosomes and female gonads, or an X and a Y chromosome and male gonads. But you can also have mice that have two X chromosomes and male gonads, because the SRY gene was put again in somewhere else, or that have X and a Y chromosome and female gonads, because the SRY gene was never reinserted. So I'm going to show you similar type of slice series through the brain again that I just showed for sex differences. But now up top, you have, is there an impact of having an SRY gene, which means male or female gonads? And at the bottom is an impact of having a Y chromosome, given that we can separate it out. And you'll notice that most of what I showed before in terms of the classic sex differences are indeed programmed by the SRY gene, or having male or female sex organs. But there are some areas of the brain that you're seeing up here that differentiate just by whether you have two X chromosomes or an X and a Y chromosome. And you can look at some of these studies in humans as well, where of course we cannot play the same genetic tricks, and we wouldn't do it even if we could for very obvious reasons. But there are these conditions of sex chromosome aneuploidies. Now, I'm sure many of you have heard of Down syndrome, which is 3 copies of chromosome 21. The other type of aneuploidy, or supernumerary chromosome, that tends to exist are in the sex chromosomes. So the most common of these conditions is Klinefelter syndrome, or having 2 X chromosomes and a Y chromosome. But there's rarer other conditions, such as having 3 X chromosomes or having 1 X and 2 Y chromosomes, etc. And there's been an ongoing study at the National Institutes of Health where they're trying to study children and adults who have these supernumerary sex chromosomes to see what differences in brain and behavior are like. And this is a pattern of where it shows up in the brain. So in the posterior part of the brain, there's a big impact of having different number of chromosomes as well. And one of the things that's interesting is just about every one of the sex chromosome aneuploidies has higher risk of neurodevelopmental disorders as well. So they have certain associated issues in terms of endocrine development, but there's also, in Klinefelter syndrome, if I remember it right, there's around 10% of kids with Klinefelter syndrome have autism as well. So to go back to timing a little bit, of course, we can look at the developing brain, and we can look at where things are beginning to change. And again, most of the brain will develop the same. This is, again, a mouse study, seeing how the brain goes bigger and how some of the areas become more complicated. And you can begin to check when these sex differences that we know in the brain begin to occur. That's what we're seeing here. Areas in red are larger in male, areas in blue larger than female. If you pay attention to the medial amygdala here, you can see that it's bigger in males throughout this developmental time period. So it's the classic sex different area. It's programmed by this early testosterone. It's going to be there from the beginning of just after birth until adulthood in mice. But if you look over here, there's some cortical areas that only around puberty in mice begin to actually show sex differences, in this case being larger in females than in males. So it's the same thing shown in this slice series. I apologize, it's a little bit dark at the bottom, where if you look, anything in red is bigger in males. It's pretty constant throughout. And then you have some of these areas in blue that begin to be larger in female mice than in male mice that really come on after puberty. And of course, if you go back to the initial thoughts that or the initial data that we had on the differences in mental health disorders, most of the mental health disorders that are pre-pubertal onset tend to be more common in boys than in girls. Most of the ones that are post-pubertal onset are more common in girls than in boys. And here we're seeing that actually development of sex differences in the brain might be linked to some of these same factors, in the sense that certain areas are changing only around the addition of pubertal timing differences. And it looks somewhat similar in humans. If you look at the thickness of the cortex in this case, the neocortex, anything that's colored is bigger in males than in females. And this is showing it at age 10, 14, and 18. And just a very simplistic glance, you can see that the brain is a little bit bigger in boys than in girls. Again, we knew that, as boys tend to be bigger than girls. But if you look at the thickness of the cortex, almost the whole brain is bigger in boys than in girls at age 10. By the time you get to age 18, many areas show no more difference between boys and girls as well. So again, this timing-dependent difference in sex development, in brain development dependent on sex, with something happening around the pubertal transition that changes how the brains respond to further development. I'm going to go into a little bit of a sidetrack to talk about some behavioral effects as well, where if you look across behavior and sex differences in humans, there's a few things that are reliably different. So, first thing we should throw out, again, as with any of the things that I've talked about today, most of cognition is going to be just about exactly identical between boys and girls, or have very, very subtle differences. But the only two things that are very reliably captured is that boys or men tend to be better at spatial memory, and most commonly you also see some language tasks that are better in girls than in boys. And you can recapture this in mice as well, where the test that we did here was looking at a radial arm maze. So basically what you do is you take a mouse and you take food away from it for a day. So you're trying to get it motivated to find food. And then you put it into the middle of this maze, and there's 8 arms going outwards. And in 4 of the arms, there's a food reward, and the other 4, there's not. And then you can simply ask, how quickly does it find the right arm, which has food? How often does it make a mistake in terms of getting into the wrong arm or revisiting an arm where it's already eaten the food? And we can do these in these mice again, where we can try to figure out, is this sex difference that we expect due to the difference in steroids, i.e. the origin from male versus female gonads, or is it because of differences in sex chromosomes? That's what you see here. We can clearly see the sex difference where female mice don't learn as well in this task as male mice. Whereas if you look at the effect of chromosomes, having an XX and an XY, there's no difference at all. In this case, begin to explain where these origins of sex differences in spatial memory in this case are coming from. Another way to think about sex differences is in terms of activational versus organizational. So far I've talked only about organizational sex differences, which means that the sex differences are programmed at a certain period in time, usually around birth, and then they remain afterwards. But there's also parts of development and parts of behavior that depends on the current status of what sex steroids are circulating through your bloodstream. And best way to look at that in rodents is to look at the female estrus cycle, which is the equivalent in rodents of the period. And basically what you have, it's females cycle for 4, 5, 6 days or so, where they start off in a stage of having high estradiol, then it goes low for 2 or 3 days, then progesterone rises a bit, and then it goes lower. And what they've shown in rats, and which we reproduced here in mice, is that the volume of the hippocampus, one of these areas that also is responsible for spatial memory and spatial navigation, actually begins to vary with the estrous cycle in females. So that as estradiol or progesterone are high, the volume of the hippocampus is largest, and then over a period of a day or two, it shrinks a little bit. Now, the effect sizes here are very small. We're talking about a 1%, 1.5% difference in volume. So again, we're not changing the brain drastically, but there are changes with the presence of estradiol in the brain that changes the brain on an ongoing active basis. And there's an interesting way that again links back into behavior, where if you take mice and you put them onto a similar kind of task here, so again, they're deprived of food, they want to find the escape box, you can see the mouse walking around here, and the mouse has 2 choices, a right turn or a left turn. One of them will have food, one of them will have not. And there's 2 ways the mouse can solve this. They can remember that it's a right turn that gets it food, or they can look around the room and figure out what cues tell me. So go to where the curtain is bright as opposed to where the curtain is dark. You can then test them on this to see how well they do by suddenly starting them after they've learned this task on the opposite arm and seeing how they learned the task. So if they learned by making a right-hand turn, they will now make the wrong turn. Whereas if they learned by the spatial cues surrounding the room, they will make the correct turn and find the food again. What's quite interesting is that across the estrous cycle in female mice, you have these changes. These mice do equally well no matter what part of the cycle they're in, but how they solve the task actually changes with the amount of estradiol in the system. Where when estradiol is high, the female mice will solve the maze exactly the same way as male mice will, in the sense that they will use the spatial cues and they will get the right answer even if you switch where they start. But when estradiol and progesterone are low on that one day, they tend to solve it using the knowledge of, do I make a right turn or a left turn? And then they will get it wrong when we do this probe test. So again, a kind of changing cognition depending on the activational properties, so the current state of the circulating sex steroids. So this was a kind of very whirlwind overview of sex differences in the brain, the basic biology of it. So I want to touch somewhat briefly back into the mental health disorders and basically what we need to understand. Now, something to keep in mind is that we actually know remarkably little. Given how interesting and how important these sex ratios are, we really haven't figured out why they play this role that they do. So these next slides are mostly stolen from Meng-Tran Lai, who wasn't able to give this talk, so I'm to some extent giving it in his place. And I'm going to focus mostly on autism, just because that's both the field that I know best and where we have the most data. So the first question, of course, that we need to get back at is it really a male-predominant condition? I gave you the number of 4 to 1. I'll touch back on that. Clearly it is, but there's a bit of a surprise there as well. We have to understand why. Does the expression of autism differ between boys and girls, or is it exactly the same, just less common? Is it under-recognized? Is it actually that there are close to as many girls with autism as boys, but we're not as good at finding them in the clinic as we are boys, because we have our preconceived ideas of what autism would look like? Is there a female protective factor? So if there's indeed a biased sex ratio, you can look at it as boys being more vulnerable or as girls being protected. And if so, that might give us important clues about why are they protected. And then lastly, are these factors that I've talked about that contribute to typical sex-gender development, are those the key ones that begin to influence autism versus not autism, ADHD versus not ADHD, depression versus no depression. And then lastly, I won't touch on this at all, but of course, this could go back to the caregiver in terms of understanding, do these sex-gender differences impact how we treat boys or girls, or should we treat them the same? So first, briefly on the sex ratios. The overall sex ratios across many studies in autism is about 4.2, about 4 to 1 in terms of boys and girls. Now, in almost all of these cases, these are studies where we use samples of convenience, which is people who are in the clinic who are asked, do you want to participate in a study? And then on we go. Now, there's other studies that you can do which are more epidemiological valid, where you go into a community and you use some of the autism diagnostic tests or some more detailed questionnaires on every person within a certain age range, and then see how many actually would fit the diagnostic criteria. And this changes. So if you use passive ascertainment, so people who just come into the clinic, it's about 4.5 to 1 boys to girls. If you use active ascertainment, so you go to a community and you try to interview every person of a certain age, it's only about 3 to 1. So there's clearly some evidence that we're missing some people in the clinic. We're under-recognizing autism in girls as in boys. But even so, there's still a significant sex difference of about 3 to 1, even if with that taken into account. What could this be due to? The only thing we have some solid evidence on is in genetics. And one way to look at that is in terms of sibling risk factors, which actually produce some quite fascinating data. If you're a younger male and you have an older sister who has ASD, your probability of getting the diagnosis yourself is about 15–16% or so. If you're a younger female and you have an older male or a brother with autism, it's only about 4%. And in between, you have these other differences here, a younger male with brother who has autism, and younger female with older sister who has autism. So given that some of the genetics are de novo, so new mutations, and some of them are shared across the family, clearly some evidence that there's some increased genetic load, increased amount of mutations that are some ways contributing to the diagnosis in girls as in boys. You can also find that more explicitly by looking at the de novo copy number variants and other types of genetic mutations. And in all cases, they seem to be higher, especially the de novo mutations, in copy number variants in girls as in boys. Now, again, you're always stuck a bit here with the ascertainment bias, that if we're not as good at diagnosing girls as boys, especially higher functioning girls, we might be getting a slightly severer end of the spectrum in girls as in boys, and therefore contributing to some of this. So there's always some risk in trying to tease out those two factors. But certainly some evidence that there's increased amount of genetic load in females, which conversely could be spun around by saying if you have the same genetic load, you're more vulnerable as a boy than as a girl. When we go into brain differences, it's actually very poorly understood. Now, one of the things is that most of the studies we do tend to have too few participants to begin with. And of course, if you have too few participants, the number of them that will be girls, given the sex biases in the field, are going to be too small to make any strong statements. So probably the most interesting study that shows some very reliable evidence comes from the 16p11 copy number variant, where there's a large consortium in Europe and in the US looking at patients with the 16p11 deletion. And in that case, even though there's slightly more men than women or boys than girls with that mutation that were captured, the sex differences in the brain were identical between boys and girls. But again, there's much, much more work to be done to begin to understand and tease that apart. So I will end with that, and I will thank many people who have helped with this presentation, especially Meng Chuan, who's down there, as well as Jane Foster, Mark Palmer, and Armin Rasenhan from the NIH. Thank you.
Being as there's the differences in the male and female brain, in terms of the DSM, should it be different for women or for girls and boys? And is that part of the issue, you think?
I mean, in some ways, I'd almost defer that question to people who have much more precise knowledge on the clinical and diagnostic side of things. So most of the sex differences in the brain are not going to be due to the kind of factors, or not going to contribute to what we would consider classic behavioral deficits in autism or ADHD or any of the other disorders. So should the diagnoses be, the diagnostic criteria be different? Maybe, but I wouldn't say that just because there's some sex difference in the brain, that's a good argument. It's more that there some differences in presentation, which is some evidence that there are, especially there's some evidence that there's more masking in females than there is in males. But I'm not sure, we don't yet know enough to directly link that back to some of the basic sex differentiation in the brain in normal development or in what we can do in rodents or birds.
As you show progression in the development of the brains, if there are any areas of the brains if there was a way to help in that development, improve the results around ADHD and ADHD, the puberty, as it looks like it's a big peak there.
Yeah.
Is there anything being worked on that?
No, partly because we're actually very bad at influencing specific areas in the brain in terms of, I mean, even if we know in the diseases where we know that there's a certain area of the brain that's very clearly involved, there's not that much we can do about it. And so our knowledge in neurodevelopmental disorders is too poor to know exactly where we would intervene even if we could. But even if we had that knowledge, beyond some things like deep brain stimulation and surgical technique, we can't really target circuits that well. We can just target neurotransmitter systems with pharmacology. So we're again, there's this gap of knowledge about, even as we're identifying some of the underpinnings of where sex differences might lie, where diagnostic differences might lie in the brain, to be able to do anything about it is a big gap that we really can't do very much in.
This might be the same question, but this was all really interesting, but I kept thinking, so what are we gonna do about it? So do you have any hypotheses? It sounds like you don't really know what is next, but do you have any thoughts as to what are you researching in terms of what we can actually do about it? Because the information on its own isn't helpful if we don't kind of know what's next. So is there any research being done as to what we can do with this information?
So there's plenty of research being done, but again, mostly there's a big gap in terms of what we know about it. So the more clinically relevant information is at the clinical presentation end. So being able to capture some of the girls who might not currently be captured, where we could help just with cognitive behavioral therapy or whatever it might be with existing treatments, who we're not getting them to because we're not recognizing them. So that's probably the most interesting bits of research. In terms of whether it might go from the basic science, there's been a decent bit of work trying to understand whether the endocrine system actually differs in these neurodevelopmental disorders and whether that's some place where we could intervene. And there isn't much evidence that we can intervene there. So the way you go from the basic sex differences and the programming sex differences to differences in diagnosis is something that is poorly understood, but it's not the simple one that the boys simply have more testosterone. If we could limit testosterone a bit, they would do better. So that's been tried, that's been ruled out, similar with that, giving them estradiol, it's been tried, nothing has really worked, or the evidence isn't there to even try it in terms of a clinical trial. So it's more at the point of beginning to rule out some of the simple things, and then hoping that as our understanding gets deeper, something will present itself as more of a way to get there. But no, it's not useful yet, but hopefully it will be, as the more we know, eventually clues will begin to fall into place.
Yes, I was just wondering, you mentioned about how when you introduce testosterone to the male and female, that there was different reactions to the things that can happen. Now, can this be translated as well to, let's say, the ingestion of growth hormones or the hormones that are in our food right now? Could that have a similar effect on the brain, male, female? And I know it might have your expertise.
Yeah. No, it will have different effects. So, and then also a more vulnerable period where if you look at, the most work's been done around estrogen analogs, especially things like bisphenol A, et cetera, where high exposure at different times will result in different brain and cognitive development, and it'll be differently affecting boys and girls. And there were some quite interesting studies in the mostly 70s and 80s, mostly in the US, but around fish being eaten from the Great Lakes, which had very high levels of estrogen analogs, and resulting in different cognitive and sometimes even sexual organ development in boys versus girls if there's high exposure to those fish. So there is evidence there, and it's going to be differential response. And there's also, in other ways, there's interesting sex by something environmental effects, which can be remarkably different in boys than in girls. So one example is and this we know well in rats, we don't know as well if it translates to humans. If you stress a female rat versus if you stress a male rat, the response of some of the neurons in the hippocampus will be the opposite. So there's a differential response to stress, for example. So in that sense, I think it's interesting to see where you get the exact same effect versus where you might get differential effect. And if you get differential effect, it might begin to explain some of these ratios that we see. Maybe that's time for last question, and then we move on.
Just the 4 to 1 number we've heard about for years and years and years. I'm just curious with the changing diagnostic criteria when Asperger's sort of moved into the autism, did that skew the numbers? And then I guess also looking across sort of this 4 to 1, it doesn't really seem to take into account level of severity. Like just my biased observation, look at sort of, I hate to use the term high functioning, but sort of high functioning Asperger's, it really seems to even out a lot. When you go into the very severe, it seems to skew.
I think it's at high functioning for all the problems the term has, where you see this big ascertainment bias. So if you have somebody presenting with an IQ less than 70, they're not going to be missed in the clinic. Whereas if you have somebody with perfectly normal IQ and some slightly different behaviors that would lead to an autism or ADHD diagnosis, there's much more possibility for that to be masked or for that to not follow a stereotype presentation pattern that's easily caught. So I don't know how those numbers have changed with the DSM-4 to 5, but certainly if you go back to the DSM-4 days, Asperger's was thought to be about 9 to 1 as likely in boys as in girls, versus autism, or as defined at that point, more like 3 to 1, 4 to 1. So in that sense, but how much of that is ascertainment bias versus how much of that is real difference is harder to say. Should we move on with that?